Good afternoon, everybody. It's a real pleasure for me being here. It's a real honor to start this new series. And, well, I also need to thank all the people I met today. It was exhausting, but that's the way science is anyways. And it was really nice talking to you and discussing these different things, and especially what you guys have here is very special from a research point of view. The translation between basic science and clinics and vice versa is outstanding. And yes, Judy said, I'm based in Zurich, and what I would like to discuss today is somehow related to the fetal origin of disease concept. And this concept simply proposes that adverse influences, mostly environmental, during early development can induce permanent changes in physiology and behavior, which then could increase disease risk in adulthood. Or if you want to put that in more provocative words, I'd like to discuss with you how the first 9 months shape the rest of your life. And one of the factors that I have been interested for a couple of years now, and I know that some of you as well, is maternal infection and immunity. So how could maternal infection during pregnancy or altered immune response during pregnancy change the trajectories, developmental trajectories, and increase the susceptibility to lasting behavioral disorders? And this is obviously not a new idea or topic. It probably attracts most interest always when we talk about very overt anomalies that you can easily diagnose. For example, we know since the 50s that maternal infection with rubella virus can cause what is known as the congenital rubella syndrome, or CRS. And CRS is characterized by very overt physical anomalies like deafness, blindness, cognitive disabilities, and so forth. But maybe a newer sort of a boost or an increased interest in this topic was seen based on the recent outbreak of Zika virus, so which has been made responsible for the incidence in babies with microcephaly. Now, this slide here somehow summarizes, or try to summarize what we know nowadays, or at least what we think we know nowadays with respect to maternal infection or altered immune response during pregnancy and long-term neurodevelopmental changes. So obviously epidemiology tells us that prenatal exposure to infection and or immune abnormalities, I come back to this, can increase the risk of neurodevelopmental disorders. And quite interestingly, this association does not seem to be pathogen specific. So a number of pathogens have been implicated, including viral, bacterial, protozoan pathogens have all been implicated in this association. In addition to that, a number of immune abnormalities, for example, increased expression of inflammatory factors such as cytokines, chemokines, acute phase proteins during pregnancy, or autoantibodies, that means antibodies that react to self-proteins, have also been associated with neurodevelopmental disorders. So it's quite a rich repertoire of immune abnormalities that are now implicated in this association. Another point I would like to mention is that immune changes in early life do not seem to be disease-specific either. So associations have been made for a number of neurodevelopmental disorders, mostly schizophrenia, autism, intellectual disability, and more recently also bipolar disorder. And that, to me at least, does not seem to be surprising, given that these disorders here share overlapping symptoms and common risk factors. Even though human epidemiology is very important to all of us who do basic research and also to inform us about clinical or risk factors that have clinical importance, there's also some criticism. And apparently nowadays, still the evidence for these associations, according to some researchers, is not solid enough. And what we do in our own laboratory, we try to boost this evidence using animal models. And that's what our lab does. And I know that some of you are very strong in that field too. And I just want to lose a couple of words with respect to animal models. So what can animals tell us with respect to infection, immunity, and neurodevelopmental disorders? So let me start with this. What can human epidemiology tell us? So it could tell us something about candidate risk factors and obviously also the clinical relevance. But human epidemiology falls short in determining causality. So do we talk about causal effects here or bystander effects? Human epidemiology is also limited in uncovering the molecular and cellular mechanisms, and also animal models here are also very useful, if not mandatory, to screen for new treatments. And obviously, and that's why this place here is great so much, because that transition here happens at this particular place, and it's a very uncommon thing to do, that you guys can connect from the clinical to the basic research side and vice versa. So to study infection, immunity in pregnancy and risk for neurodevelopmental disorders on an experimental basis, a number of animal models have been developed over the recent years. And the commonality between these models, mostly established in rodents, rats and mice, and more recently, thanks to, again, UC Davis, but the commonality is that animals during specific stages of pregnancy are challenged with a certain infectious agents or immune activating agents. And then we do study brain and behavioral development in the offspring. Now, as I mentioned, there's a couple of models that have been established over the recent years. And one of the first classes of models looked at exposure to real pathogens, such as influenza virus, toxoplasma, and more recently, Zika virus. So these models here are excellent tools to study the effects of exposure to specific pathogens that have been implicated in human epidemiology. A sort of second family was then developed to look into innate immune responses more directly. And one of the driving objectives was that a variety of infectious agents have been implicated. So one of the current hypotheses is that it's sort of the innate immune response, or part of the innate immune response, that is critical for mediating the association between infection and during pregnancy and long-term abnormalities. So these models here make use of specific innate immune stimulants, such as LPS, which is lipopolysaccharide, which is a bacterial endotoxin, or poly-IC, which is a virus-like acute phase response stimulant. I will talk about poly-IC in more detail because that's the model we use in our own lab. Or these models, a recent refinement is that these models also look into exposure to specific cytokines, and IL-6, interleukin-6, have been discussed for many years to be a critical mediator, and more recently IL-17A. So you see there is a rich repertoire already, and more recently, thanks to efforts here made at UC Davis, we have two other models that we can use to explore the association between prenatal immune activation or immune dysfunctions and neurodevelopmental disorders. One of these models is exposure, or maternal exposure, or I would say maternal allergic asthma, or autoantibodies. I mentioned that already. Autoantibodies are antibodies that can cross-react with self-proteins, mostly fetal proteins, and therefore induce some neurodevelopmental abnormalities. And these two models, even though they're a bit more recent, at least the allergic asthma model, I'm sure they will produce a number of very important data in the future. So you see there is a rich repertoire of models, and in my opinion, it's not one model that is the best model, but we could use those different models to find overlaps, commonalities, but also to look into specific effects, so whether specific pathogens would induce specific long-term abnormalities, or, as I just mentioned, we could also see whether different models produce common effects and see whether we can find an immune factor that is critical or most critical in this association. Now, as I mentioned, in our laboratory, we use a model that is based on maternal exposure to poly-IC, obviously in comparison to vehicle control treatment. We typically use pregnant mice, mostly the C57 Black 6 strain, and in many of our studies, we do compare infections or immune activation at different gestational timing. And I will show you that timing of infection matters in terms of what effects we observe. And so what is poly-IC? Poly-IC is a synthetic analog of double-stranded RNA, and double-stranded RNA is fairly specific to viruses, and double-stranded RNA is recognized by a special pathogen recognition receptor known as Toll-like receptor 3, and Toll-like receptor 3 would then induce mostly a set of cytokines known as type 1 interferons, or if you want, antiviral interferons, and in addition, a number of inflammatory cytokines. So all in all, administration of poly-IC to pregnant mice, in this case here, is a very simple but effective tool to induce a viral-like acute phase response. And as already mentioned, I'll show you some data with respect to administration of gestation A9 or 17. Gestation A9 in the mouse is roughly their mid-pregnancy, so they have a gestational period of 21 days. But with respect to human pregnancy, that would roughly, well, it depends on what cellular component we're looking at, but would roughly be at the end of the first trimester for most developmental programs, whereas gestation A17 would roughly correspond to middle to the end of the second trimester. It's very difficult to compare, but that's the rough translation to human pregnancy. So here you see just a typical cytokine response to maternal poly-IC treatment. So these are plasma levels, and please note these are logged data here, so the effects are actually stronger than they appear here. And here what is underlined in blue level, these are baseline levels, and you see typically that cytokine increase massively, and most of them peak quite early, so depending on the administration route, and we use an intravenous injection route. So what you see here is this massive increase in IL-6. You will, if you look at the hours post-treatment, you will see that some of these cytokines will go or start going back to baseline, whereas some do not during this period that we measured here. So for example, IL-10 still up a bit longer than for example, other prototypical pro-inflammatory cytokines such as tumor necrosis factor alpha or IL-1 beta here, that all go back to or relatively back to baseline shortly. This is just an idea, and we might have time to discuss the contribution of cytokines, but this is something that those of us who study these models or use these models, this is something that we need to compare. Maybe at the end of this talk, we might have time to discuss the contribution of cytokines. But this is just to show you sort of the effectiveness of the model, that we do see this cytokine increase, and it's sort of a temporary response, and it roughly goes back to baseline within 24 to 48 hours, depending on what you look at. All right, so as Judy mentioned, my original training was in behavioral neurosciences, and still I'm very much interested in the behavioral outcomes of these manipulations. I would like to show you some of the long-term behavioral outcomes. And obviously, before I'm going to show you some data, I need to mention this. It's very challenging to model or mimic behavioral abnormalities that are relevant to a human psychiatric condition. So what we cannot do in a mouse is we cannot create a mouse with schizophrenia or bipolar disorder or autism, but what we can do is we can deconstruct a complex syndrome into individual phenotypes, behavioral phenotypes, that are also relevant to a given disorder. And that's what I'm going to show you here. Namely, I'm going to show you 2 or 3 typical phenotypes that are relevant for neurodevelopmental disorders. And I'd like to start with this one, namely prepulse inhibition. And pre-pulse inhibition is based on the startle reactivity or startle reflex. So if you hear a loud noise suddenly, so a pulse stimulus, you would startle. That's a normal reflex, and you would just show the startle reaction. But if there is just a weaker acoustic stimulus preceding the loud pulse stimulus, then what happens is your startle reaction typically is reduced. So in this case, the presence of the pre-pulse here would reduce, before the pulse, would reduce the reaction to the loud noise here. And that difference is known as pre-pulse inhibition. So your startle reaction is lower if there is a pre-pulse before the pulse as compared to situations where you only hear the loud acoustic pulse stimulus. And why is this important? It's important for various reasons. Most importantly, we see reduced pre-pulse inhibition in a number of neurodevelopmental disorders, mostly schizophrenia, some forms of bipolar disorder, and also there is some evidence that autism spectrum disorder is also, patients with this disorder also show reduced pre-pulse inhibition. And the other reason is it's a very nice translational paradigm. That means you could measure pre-pulse inhibition in human subjects, for example, by the eyelid reflex, and you can do the same test in a mouse where you just measure the whole body startle reaction. So it's a very validated, nice cross-species translational measure. And what you see here is, we have done this many times, but I show you some unpublished data because I like, well, from now on, well, I can't do that here, but this is new data, but it shows you the mean person PPI, so how much pre-pulse inhibition you show, and these would be the control animals. So here, prenatally vehicle exposed animals, and these offspring here are those who were born to mothers treated with 1 milligram poly-IC, and you see there is no difference here. And those in blue here, you see these are adult offspring of mothers treated with a higher dose of poly-IC, so 5 milligram per kilogram. So it shows you here that pre-pulse inhibition is affected in this model, but it sort of is dependent on the immune stimulus intensity. And on this slide here, I would like to mention, so this is some older data here, and here we used, I show you the 5 milligram per kilogram data, but what it really shows you is that pre-pulse inhibition deficits in comparison to control treatment is seen if the immune activating or the immune challenge takes place earlier in mid-gestation, at least in our hands. Whereas here, if you do the same manipulation in late gestation, we do not see those effects. So it shows you that timing is important as well. So it's not only a dose a dose issue, but also it seems to matter when the immune system is activated during pregnancy in terms of the long-term outcomes in the offspring here. Now, another test I would like to show you is a social approach or test for social approach behavior. And there's a variety of possibilities how you could measure social interactions in rodents. And luckily, rodents are quite social animals, so it's very natural for us to measure these interactions. So what happens is, if you would have a mouse and you would expose that mouse to, so you wouldn't see the test mouse here, but you would see here an unfamiliar mouse. What happens is that your test mouse would go and explore that unfamiliar mouse, right? And so you could measure that in a simple test, and we use this modified Y maze. And on the one side, you would have this unfamiliar mouse, and on the other side, you would have a dummy object or an empty cage. And what you do is you would measure the relative time your test mouse would be engaged in social approach behavior here versus the innate dummy object. And obviously, we run this test in relation to neurodevelopmental disorders because there's social deficits is a core symptom in autism spectrum disorder, but also highly relevant for schizophrenia. Now, what you see here is the person time spent with an unfamiliar mouse. That means how much relative time your test mouse would be spending, be engaged here in this side versus this side. And you would see the chance level here. So here in gray again is our vehicle control offspring. You see a normal mouse or a group of mice would then show a clear preference to an unfamiliar mouse. And what you see here, in both conditions, in both prenatal treatment conditions, we see a reduction in social approach behavior. It's slightly stronger in the 5 milligram per kilogram group compared to the 1 milligram group, but it's not significantly different between these two conditions here. So it's an outcome here that is not so much influenced by the immune stimulus intensity prenatally, but clearly what it shows you that prenatal immune activation can reduce social approach behavior. And interestingly also that we see the same deficits or even stronger deficits if the immune activation takes place in late gestation. So it appears that social approach behavior is very sensitive in terms of manipulations, both in terms of immune stimulus intensity and also in terms of timing. So both immune activation in early or late gestation here can reduce social approach behavior in the resulting offspring at adult age. So to summarize this, obviously I just picked two examples, and over the last 10 years there's been a lot of efforts in characterizing the long-term effects of prenatal immune activation. And here is just to show you that these manipulations can lead to a wide spectrum of behavioral and cognitive abnormalities, starting from sensory motor gating. I showed you that one in terms of prepulse inhibition, but also attentional problems, social behavior we discussed quickly, exploratory behavior can be affected, and also a lot of cognitive issues can appear, especially if the immune challenge takes place in late gestation. But what I want to show you here in this slide more is so that we see similar effects using different immune activating agents. So I just picked LPS because it's been characterized quite well as well. So you see that sensory motor gating and social behavior, exploratory behavior can also be reduced by prenatal exposure to LPS or by prenatal exposure to specific cytokines, and I just picked IL-6. I didn't update it with respect to other cytokines here, but I'm sure that this list is probably, first of all, not exhaustive, and secondly, it's already outdated, so it's showing you the speed of, or the recent developments that we've seen in this particular field. But it tells you that there is at least some overlaps between the outcomes if you use different pathogens. But what I don't show you here, and it's also important to keep in mind, not everything is the same. So there is some overlaps between different immune activating agents, but not everything is really the same. But maybe we can discuss that also at the end of the talk. Now, what we try to understand now is how is this signal here translated into long-term abnormalities? So how does infection or immune activation in prenatal life translate into long-term abnormalities? And one of our recent interests is epigenetic modification. So what is epigenetics in general? So epigenetics refers to processes that can regulate gene transcription or transcriptional activity without changing the DNA code itself. And so we were wondering whether this manipulation here in early life could lead to epigenetic modifications, and maybe that could be a process or a mechanism whereby this early insult here can modify long-term functions, brain functions and behavioral functions. And there is a variety of epigenetic processes or mechanisms, and well, there is, for example, histones. Histones are these proteins that enwrap the DNA. There's DNA methylation, microRNAs. I do not want to go into detail here. I just want to mention that there's a variety of epigenetic processes that can influence gene transcription. What we are doing at the moment is we focus on one particular process, namely DNA methylation. So DNA can be methylated at CPG regions, and that's what we are interested at the moment in most particularly. So we started with sort of an unbiased approach, and the simple question was, does this manipulation in early life lead to long-term epigenetic changes in terms of DNA methylation? So here I show you some recent data from offspring samples from the medial prefrontal cortex, and this is a genome-wide capture array sequencing approach whereby you measure DNA methylation genome-wide, so each single region, genetic region. And you can see here by the hierarchical clustering, indeed, you see a lot of changes in a number of CPG regions. And interestingly, you see both hypomethylated as well as hypermethylated changes. So this is the effects if you induce immune activation in early mid-gestation, so gestation day 9. And interestingly, we also see changes, even a bit slightly more changes, if the manipulation takes place in late gestation. But if you look into the regions, it becomes clear that the genomic region becomes clear that there is hardly an overlap between the two timings. So, for example, in the early infection condition, we see about 2,400 CPG regions that are differentially methylated, and roughly 3,300 in the late gestation condition, and only about 300 are the same. So mostly they produce quite different epigenetic changes. And then when this is a long process, and I can only show you some data here, but when you look into the overlap, what happens in the overlap here, what you see here is these are genes or CPG regions in genes that are highly involved in neuronal functions. For example, neuron projection, neuron cell body functions, and so forth. And if you do some more pathway analysis, you will see that those genes that are similarly affected by the early and the late gestational manipulations, they would affect genes that are highly involved in neural development. For example, neuroligin and neurexin 2 and so forth. Now, given the changes in DNA methylation, one question is also, would that translate to changes in gene transcription? And that's always an important question to ask, because without sort of a functional output of these epigenetic changes, it's sort of we wouldn't understand what it really means. So here I give you an example of where this is actually the case. So here you see the percent DNA methylation of a particular gene, namely midline 1. And you see here in both the early infected and late gestationally infected animals, you see more DNA methylation is associated with reduced gene transcription of this particular gene. So, and that's sort of the common assumption that more DNA methylation would lead to reduced gene transcription. You could also see the opposite, namely here is Win3, and this is only affected in the offspring who were exposed to immune activation in early gestation. So here, this gene here was found to be hypomethylated, and that would translate into increased gene transcription. But it's not so easy always. And for example, here is the neuroligin data, and what you see here, we would see reduced DNA methylation, but at the same time, we see reduced gene transcription. So now the idea is, depending on where DNA methylation happens, be it a promoter region, a transcriptional start site, an intergenic region, exon region, and so forth, you could affect gene transcription differentially. And that's still, we're just at the beginning, to scratch on those data. But the proof of principle is we do also see gene transcription in those genes that we see differential methylation, even though that the changes, it's not always so easy to predict. Now, I would like to talk about something that really is highly debated at the moment, namely, given that we can induce long-term epigenetic changes in the brain, would that also mean that some effects can be transmitted across generation? So transgenerational epigenetic inheritance means that you could actually pass on a certain phenotype without a genetic basis. So, and this is highly debated in a number of pathological conditions. I give you an example. Maybe you're familiar with this, the Dutch famine cohort, which is a cohort of offspring who were born to mothers who got starvation during pregnancy during the Second World War, because the Nazis sort of isolated this region in the Netherlands. And so the first generation cohort showed a number of changes, both neurodevelopmentally, but also metabolically. And now what is happening is that now there's a second generation available to study some effects, and you can still see some of the metabolic changes in the second generation. And so that's a human example, but there's also evidence for epigenetic inheritance in other disease models, such as exposure to stress during pregnancy and so forth. So we wondered whether that's also true in this model of prenatal immune activation. And to study that, so here is I hope you can see this, it's a bit of a busy slide, but it's sort of our experimental setup we started with. So here would be our control line, if you want, so the control mothers receiving vehicle control solution that would produce a first generation control line, and some of the littermates went into behavioral testing, whereas the behavioral naive littermates were then used to produce a second generation. And the same is true for the poly-IC exposed offspring, so we'll produce a first generation poly-IC cohort. Some of them are used for behavioral testing, and the other one would be used to breed and generate a second generation. Here you would see, now I just used these two examples I was showing you before, namely, here is the social approach test that I showed you before, and this is the outcomes again in the first generation. So you see again this reduction in social approach behavior. So this is the first generation poly-IC offspring compared to control offspring. And when you look into the second generation, almost the same picture appears. So still in the second generation here, that means offspring born to F1 poly-IC animals, even without having a sort of second hit, even without having any manipulation, these offspring show social approach deficits. So this is an example of that one of the phenotypes can actually be transmitted into a next generation. And I would like to take another example where this happens, and we haven't talked about this, but this is a so-called classical fear conditioning paradigm, whereby you would on the first day, namely during the conditioning day, animals would learn about a specific association between an initially neutral stimulus, the tone or the CS, and then that is paired with an unconditioned stimulus, in this case a foot shock. And what happens is if during conditioning the animals learn to predict the occurrence of an aversive event just by presenting the tone. And what they do, rodents, if they cannot escape, they start playing dead or they freeze. Now here's the effects in the first generation. Here during conditioning, so you see that this freezing behavior increases as a function of CSU trials. So the more pairings they get, the more they learn. And here is the outcomes on the fear expression day, so on day 2, where it just exposed the tone without delivering the shock, and you measure the so-called conditioned fear response. And what you see here is we see increased fear expression in the first generation offspring of poly-IC treated mother versus control offspring. And I picked this example also because this effect is also present in the second generation. So it's another example where a phenotype that is present in the first generation is then transmitted to the next generation. Now I would like to, so not everything is transmitted, to make a long story short. So here's an example where it's not being transmitted. So here's the pre-pulse inhibition phenotype. So here's the first generation phenotype. You see the reduced, well, let's just look at this bar here, reduced pre-pulse inhibition in the first F1 generation. And when you look into the second generation offspring, it's not there. So pre-pulse inhibition apparently is not behavior if transmission takes place via the paternal lineage, but not so much via the maternal lineage. So it appears that this transgenerational inheritance is via sperm cells or anything that is via the paternal lineage, but not so much via the maternal lineage. Same is true in the fear conditioning, so we also see this in terms of fear expression. You see something going on here, but it was far from significant. But here, via the paternal lineage, we see this transmission across generation again. And then finally, we keep continuing these lines, and so we also evaluated the third generation to see how stable is this. And if anything, well, not in this particular case, but in other cases, it's astable, and it's actually even getting bigger. So here is the third generation, and I'm showing you here the paternal lineage. You see it's clearly there, so the social approach deficit you can still see in the third generation offspring here. And the same is true for fear conditioning deficits. They're still present in the third generation. Now, given these findings and other models and also epidemiological studies that look into epigenetic inheritance, maybe I would like to make this comment that, whether we should sort of redefine or rethink heritability. Nowadays, heritability, and this is just two examples from biological psychiatry, just two recent studies looking into heritability of diseases or particular phenotypes, and it's mostly still defined based on genetics. But maybe heritability goes beyond genes. It's still very debatable whether that's the case and how relevant it is, but clearly some of the models suggest that you can transmit certain phenotypes without changes in genetic code, so in terms of epigenetic transmission or epigenetic inheritance. Now, in the last, say, 10 minutes, I would like to discuss the following, namely what we haven't discussed so far is that infection or immune activation or immune dysregulations during pregnancy are quite common, and still most offspring do not show an obvious outcome, whereas others would then go into certain pathologies, for example, schizophrenia, bipolar, autism, and so forth. And obviously, what is the reason for that? And what we have so far, or what I have so far neglected, is the contribution of the genetic background as well as other exposures. And if you look into the human literature, you'd find a lot of you find a lot of evidence for gene-environment interactions, for example. So this particular study here, I'm not going to read that, but as a sort of a summary is, there is human evidence showing that infection during pregnancy can be magnified by a certain genetic liability. So here is the effect of prenatal exposure to pyelonephritis, which is sort of a kidney infection, and this increases the risk for psychosis, particularly in those with a family history of psychotic disorders. So it's taken as an index of gene-environment interactions. And how could we study that in animal models? So there's sort of two different ways we can look into that, and I just picked one example here from UC Davis, namely from Paul Ashwood's group. So we could look into, we can compare the effects of prenatal immune activation in different strains. And that's what has been done already in respect to autism-like behavior. So for example, you could look into the effects of prenatal immune activation in C57 animals versus BTBR animals. I should mention that BTBR animals per se are used as a sort of genetic model to study autism-related phenotypes. And I just picked two examples here. What you see is the effects of poly-IC in C57 in terms of sociability, and here in BTBR animals. And you can clearly see, even though it's not as it's additive effects, but you can see that you could still worsen the social interaction deficits that are present naturally in BTBR animals with prenatal exposure to poly-IC. And here's another example where you can clearly appreciate sort of the additiveness of effects. So here would be a measure of behavioral stereotypies, which is also highly important for autism-related disorders. And so see, here is the effect of prenatal immune activation in BLAC6, and here is the BTBR animals, and you see it still increase that effect in the BTBR strain. So that's one way to look into gene-environment interactions in animal models, and the other way would be to look into particular genetic modifications. Here is just I picked this one because it was one of the first interaction studies that was conducted using the poly-IC model. So in this case, animals with a mutation in this gene called DISC1, or disrupted in schizophrenia 1, was compared to wild-type animals with no mutation. And what the authors found is that prenatal immune activation interacted with this genetic variation or manipulation to produce phenotypes that are not really there in wild-type animals. So the interaction of the genetic background and the immune stimulus during pregnancy produced new phenotypes that were not seen in other conditions. And I think that's highly relevant for those of us who try to understand the neurobiology of complex psychiatric disorders, and just try to model single factor happenings such as infection alone and so forth. We should think of these complexities and try to model those complexities by such combinations here. What we personally have been interested in is how maternal infection could interact with other environmental factors, and we were interested in psychological trauma because it's another risk factor mostly for psychotic disorders. And to study these two factors, we came up with this model where we injected pregnant animals with a low dose of poly-IC to mimic a sub-threshold dose or vehicle, and some of the resulting offspring were then exposed to a number of stressors or left non-stressed during puberty, and then we would look into the long-term effects, so after a stress-free period, if you want, in adulthood. And this graph here is, well, one example of the long-term outcomes in terms of pre-pulse inhibition. So let's just focus here maybe, and you see that stress alone in puberty, at least under these conditions, did not produce pre-pulse inhibition deficits, and prenatal poly-IC exposure didn't do, at least at this dose, didn't produce these deficits. But then the combination of the two led to these synergistic effects. And we were also able to observe such additive and also sometimes synergistic effects in other measures that are relevant for psychotic disorder. Here is an example of amphetamine sensitivity. And amphetamine is a dopamine stimulating agent, and it was done in relation to the dopamine abnormalities that we see in psychotic patients. And just to make a long story short, what we see here again is that the offspring that have been exposed to the two manipulations show this massive increase to amphetamine, at least in terms of locomotor behavior. Now, I also picked this finding here because there's a lot of interest in immune system dysfunctions, either when the animals are adult or when they become adult, or when you translate that to human conditions. The idea is, are neuropsychiatric disorders associated with permanent immune dysregulations? And we looked into microglia because it's one of the immune competent cells in the brain parenchyma. And what we see here is that, again, the double hit animals show this increase in some activation markers, although I shouldn't use the term activation markers because it's probably the term activation means more or less nothing, because activation can go in a variety of different directions. But nevertheless, what you see here is that when you look into specific markers that are expressed by microglia or certain microglia with a certain activation status, you see this increase in expression of markers, suggesting that the two environmental exposures led to some sorts of microglia abnormalities. And we think now that maternal immune activation can somehow prime these microglia cells, and they're just sitting there in a state, and without another unmasking event, such as, for example, stress exposure. But I suppose there are also other unmasking factors that you would probably see the same. With that, the combination of the two can lead to abnormal microglia activity during certain stages of brain maturation. And maybe abnormal microglia activity during certain stages of brain maturation could disrupt behavioral or cognitive development and play a role in the etiology of a number of neuropsychiatric disorders. So we haven't really been able to test this hypothesis yet, but we have performed one experiment that we tried to sort of approach this answer. And by this simple experiment, what we did is we exposed animals during this second hit, so during stress exposure, we gave a subgroup of animals minocycline or vehicle control, and minocycline is a broad spectrum antibiotics, but why we picked this drug is because it's known to inhibit certain microglia overactivation statuses. For example, it's used in a number of neurodegenerative disorders. It's also clinically used in inflammatory disorders and so forth. I should also mention that it's not a specific microglia inhibitor, if there is any, by the way, but it was a first approach for us to see whether if we could reduce microglia activation during this period here, whether we could also sort of prevent the long-term outcomes in this model. And so this graph here shows you just as a proof of principle that yes, minocycline treatment is indeed effective in reducing signs of microglia activation. So here is the number of CD68 positive cells. What you can clearly see is the double hit animals that got vehicle only show this increase in microglia marker expression. And if you give them minocycline during just a period of stress exposure, you could reduce these signs of microglia activation. And most interestingly, some of the behavioral abnormalities that we have observed in this double hit model can indeed be prevented. I just picked one, and I should also, for those who are interested in this, it's published recently. Not everything can be prevented, but at least some behavioral deficits that appear in offspring that are exposed to the two environmental manipulations, some of them can indeed be prevented. Now, let me just sort of finish this or my talk by asking the question whether what we do is actually relevant to human conditions. And so I give you, my answer to this question personally is yes, obviously, and I try to convince you on the usefulness of these studies by presenting this particular study. So I showed you this model where we combined a first hit, namely prenatal immune activation, with a second sort of hit during puberty, and we saw these synergistic interactions with respect to phenotypes that are relevant to schizophrenia-related behaviors. And based on that preclinical data, we started collaborating with epidemiologists in Denmark, and we simply asked the same question: would that also happen in human conditions? And so in this particular study, it was a large study involving nearly 1 million Danish individuals born between 1977 and 1998. And out of these 1 million, you would have roughly 10,000 cases with schizophrenia. And in this large sample, you would find you have control samples as well, but you would have cases that were exposed to infection alone during prenatal life, or then exposed to traumatizing events during pubertal development, or both. And what you see here, if you look at the risk, increases in risk of developing schizophrenia, what you see here, these two factors also interact in this epidemiological setting, suggesting that infection in pregnancy and then exposure to traumatizing events during pubertal life are two factors that can interact with each other in order to increase the risk of schizophrenia, similarly to what we actually have predicted with the animal model. So this was the first study looking into these environmental interactions in a large human population. Now let me conclude. I hope I was able, despite the changes in time zones and jet lag, I hope I was able to show that prenatal exposure to immune activation can induce long-lasting impairments in various brain and behavioral functions, and some of them are highly relevant to neurodevelopmental disorders. But intensity and timing matter, and maybe we can discuss that. So some of the effects we see in these models are highly dependent on the dosage of a particular immunogen or the timing. And I also believe that epigenetic modifications might be one of the molecular mechanisms whereby prenatal immune activation can induce lasting changes, perhaps even might even provide a mechanism for transgenerational effects. So apparently epigenetic inheritance needs to be considered, and the environmental history of ancestors also matters. And finally, the effects of prenatal infection can either be, well, mostly magnified, but also can be also attenuated depending on the genetic background. But there are interactions between this particular environmental adversity happening in early life and other either genetic factors or environmental factors. So how personally I see this is that prenatal immune activation is somehow a disease primer. It's not causing a disease, it's not specific to disease, but it can act as a disease primer. And depending on the genetic background or the environmental history where this takes place, you can either go into this direction or that direction and so forth. And by that, I would just like to acknowledge my current lab members and also some past contributors who have produced those data or helped producing those data here. I would also like to acknowledge various collaborators worldwide who were involved in developing this model and getting those data. And finally, I would like to thank you for your attention. I'm very happy to take questions. Thank you very much.
Thank you very much for that. Would you care to speculate on why the paternal transgenerational transmission is stronger than the maternal on an epigenetic basis?
Yeah. So I don't have an answer to that question, obviously, but we're trying to find out. And my speculation is that the oocytes are the sperm cells are less resistant to erasure during spermatogenesis. But here's the thing, maybe we would see maternal transmission if the infection takes place a little later, because the developmental trajectories of oocytes, they differ between males and females, right? So at this stage, I cannot say that it's only via the paternal lineage, but it's in this model, we do see that. And it's actually consistent with a number of other models who look into prenatal effects. It seems to be that indeed that the transmission takes place via sperm cells more so than egg cells. But whether that's time dependent or not, we don't know. And maybe, that's a speculation, it has to do with the erasure of the epigenetic code that happens during normal development. And that might be different between males and females.
Well, that's very interesting, but as a quick follow-up, then would the maintenance of the methylation change be more enduring in the sperm cells?
That is what we predict, but maybe it's not about methylation only. So we might also see similar things in microRNA, histones. We simply don't know. We have not targeted all epigenetic processes right now. We have started looking into DNA methylation in sperm cells, and they're there, and they're also present in subsequent generations. So there is something going on, but at this stage, I cannot say whether it's DNA methylation only. But we do see persistent changes in some DNA methylation codes, but maybe the effects that we see may not be related to DNA methylation. It might be microRNA, it might be histones. You know, it's a lot of possible explanations here.
Any other questions? Get back to Janine.
Yeah, as kind of follow-up to that, you showed some specific examples of genes that had changes in methylation. Are any of those maintained in the F2?
Yeah, so actually,
which genes? Or?
Yes and no. Both is true. So there is some transcriptional changes that you can follow up in the second generation. I haven't showed those data, but they're actually published. We have only published amygdala so far. We have done an RNA-seq, and you see transcriptional changes that are passed on from the F1 to the F2 and even F3. But most interesting, what interests me more is actually the changes. So some genes are differentially changed across generations. But those genes that I've showed you here, they're also present in the second generation and maybe also the third. We haven't done all of this third generation studies so far. But there are, at the transcriptional level, clearly you can transmit transcriptional abnormalities from one to the next generation.
But you can't explain it yet by DNA methylation?
So far, no. It's just one of the hypotheses, but well, the obvious thing to do would be to try to prevent those changes in the first generation and see whether it also prevented in the second generation. That's the one approach we're actually trying to do right now. And the other one is to see with artificial insemination whether we can actually see the same phenotypes by just collecting sperm cells in IFF1 and then see by artificially inseminating control females with that sperm pool whether we can induce the phenotypes that we've seen here, and then also look into methylation, gene expression, and so forth. So these are the two.
And have you ruled out a possible genetic change in the F2 or F3?
Genetic changes.
I mean, I think it'd be nice to sequence them.
Yeah. I totally agree, but we haven't done it so far. But it's likely, but rather it's possible, not likely, but it's possible that it might happen. But in 2 generations, it's rather unlikely, at least in the C57. But you're right. I mean, in the end, we need to rule out this, and also CNVs and so forth. We haven't done the homework properly in terms of what is the actual contribution. It's just a first proof of principle that you can actually transmit a phenotype that with the first exposure was in environmental hit somehow, and without just inducing another hit or a third or so forth, you can induce that. But we're at the very beginning of, delineating what processes are actually responsible for that. Yeah.
Okay, thanks.
So a little more basic, but just was there any improvement in any cognitive skill, reaction time, or is it kind of all bad news?
Well, that's a good point. So we haven't done proper cognitive assessment in these transgenerational models, I'm afraid, because logistically it's already quite large scale. But what I can tell you is that some effects actually, yes. So some depression-related phenotypes are actually getting better, or some depression-related things go into that direction what you just mentioned. But in respect to getting better, I can't say really, because we haven't really looked at it. So we have had a number of behavioral tests, a battery, but that was not really involving cognitive testing, because just it's too much of in the mouse models it's very difficult to go for cognitive testing. So I'm afraid I don't have the answer for that question, simply because we didn't do it. But yeah, it might be on the bucket list at some point. Yeah.
You mentioned there's a priming effect for microglia and also the masking effect on secondary heat. I just wonder about this. Are there any kind of long-term effect on the whole systemic immune response when got prenatal infection or immune activation?
Yeah, so if I understand correctly, what you ask is whether immune activation generally induces lasting effects on the immune system in the offspring, right?
Yes.
Yeah. Yes. The answer is yes, but it depends. It's influenced by a number of factors. First of all, age. In the brain, it's brain regions. So, for example, you see a lot of cytokine changes, but again, that depends on brain region and age. And then in the periphery, there's the research going on here who could answer that question much better than I do. But there are changes, and also in the periphery. So you can induce persistent changes. Some of them you would need to stimulate. So you wouldn't see in a basal state, but you would, for example, need to stimulate the system again, for example, by re-exposing the animals to LPS or poly-IC, and then you would see the effect. And some effects are even there in basal states, but the answer is yes. Yeah. Mm.
Where? Sorry. Oh.
Sorry if I missed it, but can you comment on whether you tested both male and female, and if you did, if you saw any sex effects in the offspring?
Yeah, you could point. I actually wanted to mention it, but I forgot. So all the data I showed you here are both present in males and females. So all I showed you here is present in both sexes. But there's a number of sex differences, and in a number of tests I haven't shown here. But what we see is that sex-specific effects emerge more so if the immune challenge takes place in late gestation. Whereas in the early gestation models, there is hardly sex-specific effects. So it's in both. Or on the other hand, you might need 400 or 500 animals per cohort to see a small difference. But when you challenge them in late prenatal stages, you start seeing sex-dependent effects, both in terms of behavior as well as neurochemistry. Yeah. Sorry, I forgot to mention that. So all what you've seen here is seen in both sexes. Yeah.
Well, thank you again very much for this very interesting presentation. I was wondering what maternal immune activation can teach us about the epidemiological trends in autism specifically, and the recent rise in incidence rates, both in the United States and probably throughout the Western world. I'm suspecting that pregnant women today probably experience less infection than it was 50 or 100 years ago due to better healthcare, vaccination, infection control, and so on and so on. But nonetheless, autism incidence rates continuously rise.
Yeah, that's
so
true.
what's the correlation there, if there is any?
Yeah. So there is, well, first of all, we don't know from human epidemiology, but what I just made what I sort of pointed out was infection, but you can have immune dysregulations from a number of environmental factors, be it diets. So obesity during pregnancy, for example, is a major contributor to inflammation, so inflammatory factors, and that increases as well. We do have, and we're actually quite interested in that, we have exposures to environmental toxins, nanoparticles, exhaust, and so forth, which all act to the immune system as well. So maybe what you see is, well, what the immune activation models tell us is maybe not that infection rates increase, and that's why, actually they decrease, what you just said correctly. Although I should mention that rubella, for example, there's a very nice study conducted in Italy with exactly this idea that we should actually have, killed rubella, and it's not the case. So even quite a number of pregnant women, I don't know the numbers right now, but there's a certain percentage of women who still get infection with rubella during pregnancy. This is an Italian study. I don't know whether that applies to the States or Switzerland, where I come from, but certainly we didn't get rid of all these things. But I do agree with you. In general, maybe 100 years back, 200 years back, that was more of an issue. But probably we also had more lethality, right? So probably more of the babies were lost, and that's a typical thing you have when you have an infection, severe infection during early pregnancy. In many cases, you lose the babies. But to make a long story short, this is the major interest has been placed on infections. But what we know nowadays is there's a number of environmental exposures that actually act on the system, including stress. I mentioned toxins, environmental diesel exhaust, nanoparticles, obesity, and so forth. And some of them increase. Some of them increase in terms of incidence. But you're right, in terms of human epidemiology, there is no clear data on that. So what is, for example, the percentage or the incidence rates in countries where you would have less infections during pregnancy? This is very difficult to study in human epidemiology simply because of the sample sizes you would need and the design of the studies, right? So, yeah.
So sort of, related to that, do you think because there was a shift in what you would take to reduce symptoms, right? So we don't take anything except Tylenol anymore, which is not a good anti-inflammatory, where they used to take aspirin, right, to reduce the fever during pregnancy. And I wonder how much that has to do with sort of the shift that we've seen. Because there was a paper that showed that women who had infection and took some kind of an anti-inflammatory to reduce fever actually reduce the risk, right? Yeah,
that's true. At least, it's in the animal models, you could do so clearly. I mean, you could actually see this dampening effect. So you just mentioned anti-inflammatories, but there's also, for example, diets during pregnancy change too. You can use diets that are anti-inflammatory, and maybe you're right. Maybe it doesn't parallel the increase in autism incidence with, it doesn't really parallel each other if you think about that. And maybe you're right. Maybe it has something to do with us, taking these anti-inflammatory drugs or changes in diets and so forth. And there is data in animal models where you can clearly see that. So you can prevent effects by, for example, giving them drugs that would reduce fever. Another issue is manipulations that would act on micronutrients, because in inflammatory states and infectious states, for example, you would have reduced iron, you would have sorts of a temporary iron deficiency or zinc deficiency and so forth. And if you would supplement the mothers with iron or zinc, you could also prevent the long-term effects in the offspring. And so it might be, so this discussion we have might be related to a number of factors, diets, drugs that we take and so forth. So yeah, it's a good point, very good point.
So one last question, Susan?
I was interested in the data that you showed about the minocycline treatment, lowering the signs of microglia activation, I guess it was.
Yeah.
But I don't think you said much about what you think the mechanism behind that is. I mean, is it MMP9? Do you have any thoughts about that? Because that could be an interesting sort of avenue for treatment.
Yeah, so I don't know, because minocycline is such a dirty drug in terms of mechanisms. It might be related to metalloproteinases, MMP9, for example. You're right. We simply don't know. We haven't followed this up because we sort of then move more into the epigenetic field. But it might give us an important indications on how this whole system works. I can't tell you because simply we did not follow this up properly. But yeah, microglia, they need the extracellular matrix, right? So it might be likely that, or at least plausible, they have something to do with the ECM, with the extracellular matrix. Whether that's the case, yes or no, I don't know. Another thing I didn't show here, what was very interesting though is, microglia activation or dysregulated activation and so forth, you could see from many angles, and we were interested in the contributions of neurons that would inhibit microglia. So for example, CD200 and CD200 receptors on the microglia. And in this double hit model, you would see quite a lot of changes from the neuronal sides. So it might also be that with a minocycline treatment, we sort of correct the neuronal problems and then would act on the microglia, sort of a vicious circle. But that's just pure speculations. I simply don't know. Yeah.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.